Hello friends, welcome back to Noah Bio. Today we are going to talk about cell cycle. What exactly happens when cells divide? Okay, that we are going to talk about. Now, when cell cycle is not regulated, when cell division is improper or incorrect or uncontrolled, that leads to cancer. That is oncogenesis basically, and that is because of some genes. For example, tumor suppression genes such as P53 is no longer working, is no longer functional. Okay, so what exactly happens? Cell division is a continuous process. Your skin cells divide throughout 24 hours. Your gut line divides throughout 24 hours. There are few cells which divide at a faster rate, few cells divide at a slower rate. Cell division is not at a constant speed. Cells which are present in a particular organ will divide faster depending on the requirement of the cells present at that particular location. Now, cell division is a bit a complex process. Now, when that process gets hampered, that leads to problems. And what are the problems? We will see that, and how our body manages to repair that, how our body manages to kill a particular cell if it is not following a particular path. That also we can learn under cell cycle. So as we can see, cells divide and they grow, they multiply over the period of time. How do they grow? We'll see as a step. Now there are two phases basically in which cell division occurs. Those two phases are divided into mitosis and interphase. Okay. Now we will see that in depth. See, mitosis is only for somatic cells. Okay, it is not for germline cells. Remember that mitosis is only for somatic cells. So we will see that. So mainly cell cycle is divided into mitosis and interphase. Okay. In turn, there are three subphases of interphase, which is further divided into G1, which is also called as gap 1 phase or growth 1 phase, S phase, which is synthesis phase, and G2 phase, which is gap 2 phase or growth 2 phase. So we will see that in depth. Now remember that what happens, there are regulatory checkpoints are there, okay, and there are cyclins and cyclin dependent kinases and tumor suppressor genes. Okay, so all of them come together and play a role of cell cycle. So if cell division is not proper, if cell organelles are not multiplied, if DNA is damaged or chromosome is damaged, then these checkpoints will come into play and they will halt the cell cycle. Okay, if that particular problem, that particular, say for example, DNA damage is there and if it is not getting repaired, then cell will be directed to programmed cell death, which is also known as apoptosis. Okay, remember that there is a difference between apoptosis and necrosis. We will not go into details of that, but if the damage is not able to be repaired, cell is not able to repair that damage, cell will be directed to the programmed cell death. Okay, now we will see the phases of cell cycle. So generally cells are in G0 phase. Okay, at G0 phase they are in quiescence. Quiescence means there will be no activity. They will be just like that. They will be laying over there just like that. Once cell wants to divide, they will enter into a cell cycle phase. So the first phase will be G1 phase under interphase. So the first phase which comes under interphase is a G1 phase. So cells are at G0 phase, they are not going to divide at all. Once they want to divide, they will enter into G1 phase. In G1 phase, what exactly happens? Cell organelles will multiply for the growth. Okay, cell organelles, remember that mitochondria, endoplasmic reticulum, Golgi complex will multiply. Why? Because from one cell, two daughter cells are going to be produced, right? Identical daughter cells. So for that sake, obviously, DNA is going to get replicated, organelles should be replicated, then and then only from one organ, two organs can be produced, right? So replication is important. So in G1 phase, organelles, remember that organelles such as mitochondria, endoplasmic reticulum, Golgi complex will multiply. From G1 phase, cell will be directed to S phase. S stands for synthesis, DNA synthesis, which also in turn replicating DNA replication. It is talking about a DNA replication. Okay, so in that S phase, DNA will replicate from one DNA double stranded, two DNA strands will be produced in S phase. From S phase, it is going to get directed to a G2 phase, which is a gap 2 phase. In gap 2 phase, the organelles doesn't divide, neither DNA replicates, but it is preparing the cell for mitosis. It is preparing a cell for mitosis. So, from S phase, cells will be directed to G2 phase, and then from G2 phase, it will be directed to M phase, which is mitosis phase. In mitosis, particular separation of chromosomes will occur and two daughter cells will be produced after mitosis. Okay, so this is how cell cycle works in terms of phases. Now it is handled by cyclin dependent kinases and cyclins. As the name cyclins, cyclins represent a generation of chemical molecule which is undergoing generation and degeneration over the period of cell cycle phase. That's why the name is cyclin. Cyclin dependent kinases. Kinases are basically proteins, a group of proteins basically, which helps in phosphorylation. So what is the role of each cyclin and CDK? Together we'll see step by step. Cyclin dependent kinases and cyclins, they together work in a combination. So when cyclin dependent kinase are phosphorylated and bound to a CDK, then cascade of mechanism acts. In between whole cell cycle, there are checkpoints. So we'll see which are the checkpoints and which are the CDK and cyclins play together to clear out that checkpoint if the cell is proper, if the division is proper. Okay, so we'll see one by one. This is the first checkpoint at G1S phase. Okay, you can see. This is the second checkpoint at G2M phase. This is another checkpoint, and this is the last checkpoint at CDK1 between mitosis and G1 phase. Okay, so we'll see how CDK and cyclins work together. First, cyclin D and CDK4 or 6 will come together as a heterodimer. They will come together, they will undergo phosphorylation. In turn, they will act on E2F RB1. After their action, E2F gets phosphorylated. Okay, remember that CDK requires ATP for phosphorylation. Over here, CDK kinase, which is cyclin dependent kinase, will in turn phosphorylate E2F. The phosphorylated E2F will go and act on cyclin E CDK2 complex for phosphorylation. If the cell is going from G1 to S phase is proper, cell organelle division is proper, then only this checkpoint will be cleared. If the concentration of CDK2 and cyclin is not up to the mark, not up to the required concentration, then this checkpoint will not be cleared. Cell cycle will halt. Cell cycle will stop at this particular position. Now, if this point is clear, if this checkpoint is clear, when the concentration of CDK2 and cyclin E is proper, then cell will move on to synthesis phase. It will undergo DNA synthesis, then it will move ahead. If the DNA synthesis is not proper, then the concentration of CDK2 and cyclin will not be enough, then cell cycle will not move ahead. If cell is able to repair the DNA concentration, if DNA damage is there, cell is able to repair the DNA damage, then the concentration of CDK2 and cyclin will be proper, then only cell will move on to next phase, which is G2 phase. At G2 phase, cyclin A and CDK1 concentration is there. If the concentration is proper, then cell will pass this phase. After this, cell will enter into mitosis. At mitosis, two daughter cells needs to be produced from mitosis. Mitosis is divided into prophase, then anaphase, metaphase and telophase. So that process will occur, and from one cell, two cell will be generated. At the end of mitosis, again there is a checkpoint the checkpoint will be handled by CDK1 and cyclin B. If the concentration of CDK1 cyclin B is not proper, cell will not go to G1 phase. Now remember that after this particular completion of mitosis, cell can either enter into cell cycle again, that is G1 phase, S phase, G2 phase and so on, or cell can go into quiescence, which will go to G0 phase and cell will not divide. It depends on the requirement of the cell. Now remember that there are inhibitors. Okay, if cell cycle is not proper, if the division is not proper, then these inhibitors, the inhibitor of CDK4 and 6 will go and bind to CDK4/6 so that phosphorylation will not occur over here, and in turn this phosphorylation of E2F will not occur and this phase will not go ahead. Then there are CDK2 inhibitors. If the synthesis is not proper, then CDK2 inhibitor will go and bind to CDK2 and in turn CDK2 for cyclin dependent kinase 2, and in turn they will not allow this phase to be passed, this from S phase to G2 phase. Okay, it will not allow by the help of inhibitors. If the growth phase is not occurring proper, if the cell growth is not proper for the mitosis, then the inhibitors of CDK1, cyclin dependent kinase 1, will go and inhibit over here, so the concentration of this will not be enough and then it will not pass from this checkpoint. Once all the points are going to get clear, if everything is happening at a proper step, in a proper manner, okay, then only cell cycle will progress. Remember that these inhibitors play a major role. Okay, if inhibitors are not there, then cell cycle will not go properly. Remember that we also have tumor suppressor genes. Okay, if cell is going continuously under cell cycle, uncontrolled manner, uncontrolled growth, then that results into a formation of tumor. Okay, that results into formation of cancer. There are two types of cancer, and there is whole cancer biology which is called as oncogenesis. In that we are having two types, benign cancer and malignant cancer. We will not go into details of that, but this is to give you idea about cell cycle. So basically just we will revise it once again. Cell cycle is basically divided into two main phases, mitosis and interphase. Okay, interphase is in turn divided into G1 phase, gap 1 phase or growth 1 phase, synthesis phase and G2 phase. Now interphase is the longest phase, okay, and G1 takes around 8 to 10 hours time. Synthesis phase is of 6 to 8 hours time, whereas G2 phase is of 3 to 4 hours time. Okay, the duration of the cell division. Now each and every cell will undergo division. Remember that mature RBCs and neurons doesn't undergo cell division, doesn't undergo cell cycle. Okay, so it is not necessarily every cell will undergo cell cycle. It depends on the requirement of cell and cell organelle. So this is to give you idea about cell cycle. If you have liked the video, please like, share and subscribe, and do comment in the comment box.